Okay, so fat mobilization. In order to get fat and use it to use it for energy, you have to get it from the fat cell into the bloodstream. So epinephrine kind of kickstarts this whole process. So it enters into the cell, and then ATP kind of kickstarts PK, takes it from inactive to active PK, which then activates HSL, and it turns triglycerides, and then HSL makes it into glycerol and free fatty acids. And glycerol can easily go into the bloodstream, or can then be taken into the liver. However, free fatty acids are not capable of doing that. They cannot just enter the bloodstream on their own. They have to be attached to a plasma protein, in this case, albumin. So then they attach, they can enter in the bloodstream, and then they can go into the other tissues. So from there, we have our free fatty acid. And this is currently in the cytosol of the cell. It still has to get inside the matrix of the mitochondria. So it has to get through this first membrane and the second membrane inside the matrix. So it does this by the free fatty acid combined with coenzyme A turns into acyl-CoA. And this is done via acyl synthetase. And in this process, you actually lose an ATP. So it's an energy investment stage, kind of like glycolysis. So ATP turns into AMP with 2-phosphates. So acyl-CoA is then combined with carnitine, which then turns into acylcarnitine, and this is done via CAT1. So acyl-CoA, carnitine, acylcarnitine. And this can actually enter in through the first membrane because it's porous, so it enters in. However, it can't get through the first or the second membrane on its own, so it actually requires acylcarnitine translocase. So that then takes it inside of the mitochondrial matrix. So then acetylcarnitine via CAT2 is then broken back down into carnitine and acyl-CoA. And from there, acyl-CoA goes through like a cyclical process, and it's a 4-step process that requires water and coenzyme A, and from there it actually turns into acetyl-CoA, which is what is needed in the Krebs cycle. In this process, so we actually convert FAD to FADH and NAD to NADH, which is useful because it can then go clearly into the electron transport chain where it can be used there. So now we have acetyl-CoA, which can be taken into the Krebs cycle, and actually that carnitine can be taken back outside the cell where it can be recycled to start the whole process over again.